Okay, hello everyone. So here we are today again continuing with our story on metabolism of amino acid, but this time we're gonna shift our focus from the metabolism of nitrogen atom to the metabolism of amino acid skeleton, and we're gonna discuss parallelly about some disorders of amino acid metabolism. So first I'm going to make an overview of the amino acid carbon skeleton metabolism, and then we're going to focus on amino acid metabolism one by one later on. So first let's list the important coenzyme in amino acid metabolism that we are gonna mention and repeat from time to time, and in order not to make digression at the time, let's list them now. So first one is pyridoxal phosphate, which is derived from vitamin B6 pyridoxine, and this one is used for removal of amino group from amino acids in the processes of transamination, and that is something that you have already heard about in our previous video. Also, it can be used for donation of amino group during amino acid synthesis, and definitely this enzyme is involved, and you will see that in the upcoming minutes, involved in certain reactions that involve carbon skeleton of amino acids. The second coenzyme is tetrahydrofolate FH4, which is responsible for transfer of one carbon fragment, which can be in various oxidation states. This one is involved in the reaction of degradation of amino acids, and you will see that there are more of them. Here I have listed just a couple of examples. For example, degradation of serine and histidine. But on the other side, FH4 is also involved in the biosynthesis of certain amino acids, for example, glycine, and you will see it in the upcoming couple of minutes. And the third one is Tetrahydrobiopterin Bh4, which is involved in hydroxylation processes, especially in ring hydroxylation reactions. And we will see the role of BH4 in conversion of phenylalanine to tyrosine. Actually, BH4 acts in a way that it requires the involvement of molecular oxygen in a way that one atom of oxygen is incorporated into the new molecule in the form of hydroxyl group, and the other oxygen atom is incorporated into the molecule of water. So what about amino acid biosynthesis? So 11 out of 20 amino acids can be synthesized in human organism, and other amino acids are considered to be essential, and they have to be taken through diet. So these amino acids can also be further involved in the biosynthesis of other nitrogen-containing compounds. And I have listed you here a couple of examples, and we're gonna go into details more in our next video. So glycine is used for the synthesis of porphyrin and purine, glutamate for the synthesis of neurotransmitters and purine, aspartate for synthesis of purine and pyrimidine, and also amino acids are used for synthesis of glutathione, serotonin, creatine, and so on and so on. So let's take a look at this figure, which represents what's actually going on in amino acid synthesis. So they can be synthesized from glucose. Because during glycolytic pathway, phosphoglycerate is formed, which can be used for the biosynthesis of serine, and serine is used for the biosynthesis of glycine and cysteine. Cysteine requires methionine as a donor of sulfur atom in a group. Also, pyruvate as the final product of glycolytic pathway is used for the biosynthesis of alanine in transamination reaction. Further on, pyruvate can be converted to oxaloacetate, and oxaloacetate in transaminase reaction can be converted to aspartate, and aspartate is a precursor for the synthesis of asparagine. Pyruvate can be converted to acetyl coenzyme A, which enters together with oxaloacetate to Krebs cycle, and some of the intermediates of the Krebs cycle, like alpha-ketoglutarate, can be used for the biosynthesis of glutamate in transamination or deamination reaction, and glutamate is used for the synthesis of glutamine and proline and arginine. So you can see that from different intermediates of carb metabolism and fatty acid metabolism, we can get certain and synthesize certain amino acids. So definitely carbon atoms in amino acids originate from glucose, and nitrogen atom is donated from either ammonia or from another amino acid. Tyrosine and cysteine are considered to be conditionally essential amino acid because for their biosynthesis, they require the presence of phenylalanine, which is essential amino acid, and methionine, which is a donor of sulfur, which is also an essential amino acid. So carbon atoms, as you can see, can be derived from glycolysis or Krebs cycle. So let's make just a list. From glycolysis, we can get serine, glycine, cysteine, alanine. From Krebs cycle, we can get glutamate, glutamine, proline, arginine, aspartate, asparagine, and so on. The regulation of this process is achieved and accomplished by feedback inhibition, which is actually exhibited in the form of allosteric modification, or it has an effect on the transcription of an adequate gene. Degradation of amino acid is different from biosynthetic pathway. Sometimes we are going to have, and we're going to see that those intermediates are the same, but enzymes are different, but in most cases we have different intermediates and different enzymes. But what is the common characteristic for all of those amino acids is that almost every amino acid can actually generate reduced coenzymes, NADH, and in most cases FADH as well. And that can be achieved either through direct oxidation or oxidation in the Krebs cycle, or conversion to glucose and subsequent oxidation of glucose, or conversion to ketone bodies and subsequent oxidation of those ketone bodies. Which of these mechanisms are going to be involved at the specific moment in the organism, that depends on the physiologic state of the organism. So during fasting periods in liver, carbon skeletons of amino acids are converted to glucose, ketone bodies, and carbon dioxide. In fat state, in liver, carbon skeletons are converted to depots of glycogen and 3-acylglycerols. So we can say that fate of amino acid carboskeleton is actually parallel to the fate of those for glucose and fatty acids. And if you notice that I have mentioned liver several times, so despite the fact that those metabolic processes of amino acid can take place in different cells and tissues like gut, like muscles, like kidneys, and so on, actually liver is the only tissue that has all amino acid metabolic pathways, all enzymes, all intermediates, and the metabolism of amino acid is rather really frequent in liver. So amino acid can be degraded to either directly form carbon dioxide, like we are going to see for glycine, or to form glucose in liver through pyruvate or Krebs intermediates, or to form ketone bodies. So this second point, formation of glucose. So let's see which amino acids are involved in specific parts of glycolytic pathway or Krebs cycle. So threonine we will see that it's converted to glycine, and tryptophan in glycine can be converted to alanine, serine, and cysteine, which can be converted to pyruvate. Pyruvate can be converted back to alanine and excreted to blood, or pyruvate can undergo oxidative degradation to acetyl coenzyme A and enter Krebs cycle, or to oxaloacetate. Aspartate and asparagine can be transformed to oxaloacetate and enter Krebs cycle. Arginine, histidine, glutamine, and proline, they tend to form glutamate, and glutamate via transaminase reaction or deaminase reaction is involved in Krebs cycle through and via alpha-ketoglutarate. Some amino acids like valine, threonine, isoleucine, methionine, they tend to convert to common intermediate propionyl coenzyme A, which is actually the same molecule which we got upon the degradation of fatty acid with an odd number of carbon atoms. And this is something that we know, that propionyl coenzyme A is converted to methylmalonyl coenzyme A and then reorganized to succinyl coenzyme A, entering TCA cycle. Aspartate, tyrosine, and phenylalanine can be degraded to fumarate, entering TCA cycle, and fumarate can be converted to malate. Malate can be used in liver for the biosynthesis of glucose, or in other tissues can be transformed to pyruvate and get ready for gluconeogenesis or whatever. So the third one, and you will see it from the next slide, it's that certain amino acids can be converted to ketone bodies like threonine, lysine, isoleucine, tryptophan to acetyl coenzyme A, phenylalanine and tyrosine to acetoacetate, and leucine to hydroxymethylglutaryl coenzyme A. So amino acids can be classified in three groups. Glucogenic amino acid, because upon the degradation of their carbon skeleton we can form glucose. Ketogenic amino acid, because upon the degradation of their carbon skeleton we can get acetyl coenzyme A or acetoacetate. And there is a group of amino acids which can be both glucogenic, C instead of K, glucogenic and ketogenic as well. So I'm going to make a list in order to prevent getting back to this list all over again when we move on to individual amino acids. So glucogenic amino acid, and this is going to be very useful for your test, are glycine, alanine, valine, aspartate, glutamate, asparagine, glutamine, arginine, histidine, cysteine, methionine, serine, and proline. Ketogenic amino acids, purely ketogenic and exclusively ketogenic, are only two amino acids, leucine and lysine. And glucogenic and both glucogenic and ketogenic amino acids are isoleucine, phenylalanine, tyrosine, tryptophan, and threonine. So they can be both. So they can synthesize glucose or they can synthesize ketone bodies and acetyl coenzyme A. So let's start one by one. First, we are going to discuss about the metabolism of amino acids via intermediates of glycolysis, and 4 amino acids are actually metabolizing through those intermediates: serine, glycine, cysteine, and alanine. So let's go one by one, and this is an overview of the pathway. So serine, so this is the first one. So these processes of synthesis and degradation of serine are especially intensive in liver and in the kidney. And actually the biosynthesis of serine starts from 3-phosphoglycerate, which is a glycolytic intermediate, and 3-phosphoglycerate actually undergoes oxidation of hydroxyl group to keto group by 3-phosphoglycerate dehydrogenase, and of course the acceptor of reducing equivalents is NAD coenzyme, converted to reduced form of NADH. 3-phosphohydroxypyruvate, which is formed as an intermediate, undergoes reversible transamination reaction with glutamate and alpha-ketoglutarate pair in the presence of course, pyridoxal phosphate to form 3-phospho-L-serine. So instead of keto group, instead of keto acid, we now have amino acid. And of course, we have to remove this phosphate group by phosphoserine phosphatase to produce serine. Here we have an example of feedback inhibition. So serine, which is the final product of this biosynthetic pathway, inhibits both phosphoserine phosphatase and 3-phosphoglycerate dehydrogenase. Serine is catabolized in 2 ways. The major pathway is actually, again, one transaminase reaction by the pair pyruvate and alanine, and in the presence of pyridoxal phosphate to hydroxypyruvate. So again, amino group is eliminated and transferred to pyruvate, and hydroxypyruvate is formed. And then we have the reduction of keto group to glycerate, and we, of course, the donor of reducing equivalents are NADH coenzyme. And then we have phosphorylation, to 2-phosphoglycerate, which is a glycolytic intermediate converted to phosphoenol pyruvate and subsequently to pyruvate. Of course, there is one alternative pathway of serine degradation, which is the beta elimination of hydroxyl group to form pyruvate directly. So not via these intermediates, by directly to pyruvate. Speaking of glycine, here is the figure explaining the biosynthesis and degradation of glycine. So if you can see from the figure, glycine can be synthesized from serine, and this is the major pathway of the biosynthesis, or threonine, which is the minor pathway. And this is an aldolase-like reaction because of the presence of hydroxyl group on beta carbon atom. So acetaldehyde is released from the system, PLP is required, and glycine is synthesized. But the major pathway is glycine synthesized from serine, where one carbon fragment is transferred to FH4, and also apart from FH4, PLP is required in serine hydroxymethyl transferase reaction. This is the biosynthesis of glycine. But speaking about the degradation of glycine, we have two pathways. So the major degradative pathway is actually oxidation and deamination of glycine to glyoxylate. The reaction is catalyzed by D-amino acid oxidase, so we need molecular oxygen for the reaction. Hydrogen peroxide is formed, ammonia is released, and we have the formation of glyoxylate. So this degradative pathway is clinically relevant because glyoxylate can be further oxidized to form oxalate. And you know that oxalate tends to form precipitates with calcium ions, calcium oxalates, and this is the basis of the formation of kidney stones. Approximately 40% of kidney stones are of oxalate origin. So there is the reversible reaction where glyoxylate can be converted back to glycine by transaminase reaction, but this is not considered to be the pathway for the biosynthesis of new glycine. Glycine can be further degraded to carbon dioxide directly by glycine cleavage enzyme. Reducing equivalents are accepted by NADH, and one carbon fragment is accepted by FH4. The lack of this transaminase actually yields to the accumulation of glyoxylate, and glyoxylate, of course, spontaneously tend to form oxalates, and this is disorder marked as primary oxaluria type 1. Regarding cysteine, the story is pretty complicated and complex. So carbon atoms, as you can see from the figure, and nitrogen atom, they originate from serine molecule, and methionine donates only sulfur atom. Because of this donation of sulfur atom from methionine, cysteine is considered to be conditionally essential. So cysteine is synthesized upon the reaction of serine with homocysteine, which is derived from methionine, in the reaction which is catalyzed by cystathionine beta synthase. And here we have one cystathionine molecule, which is further hydrolyzed by cystathionase to cysteine and alpha-ketobutyrate. Alpha-ketobutyrate is then decarboxylated to propionyl coenzyme A, and the fate of propionyl coenzyme A is very well known to succinyl coenzyme A. So cysteine is further can be catabolized to form cysteine sulfinic acid by oxidation of SH group, and further on cysteine sulfinic acid can be transaminated to form pyruvate, and sulfate is released, which is oxidase to sulfate. And sulfate is excreted either in urine in the form of sulfate, or it's converted with ATP to so-called PAPS, which is 3-phosphoadenosine 5-phosphosulfate, which is an activated form of sulfate used for a numerous biosynthetic pathway of the molecule which require sulfur atom in their structure, like some glucosaminoglycans or something like that. This is at the same time, this is the major degradative pathway, and this base is methionine, homocysteine, cysteine, and this is actually the only degradative pathway of homocysteine. So regarding the fact that all these enzymes and step require pyridoxal phosphate as a coenzyme, the vitamin B6 deficiency yields the formation of homocysteinemia, which is associated with cardiovascular diseases. But we have several other amino cysteine defects. Cystinuria is a disorder which arises from the deficiency of cystathionase, so this phase of the reaction, which also requires pyridoxal phosphate, so it's connected to the vitamin B6 deficiency. But this disorder is not followed by characteristic clinical abnormalities. And this cystinuria is relatively common in premature infants. Then we have another disorder, which is cystinuria, and this is the defect of transport protein for cysteine and cysteine. And cysteine is not a very soluble molecule, so it tends to form renal calculi. Cystinosis is a disorder arising from cysteine accumulation in lysosomes, and it tends to form insoluble crystals in lysosome, and at the same time it actually yields the depletion of intracellular cysteine, and in children between 6 and 12 years it yields the renal failure. It leads to the formation of renal failure. Alanine, this is what we all know, it can be synthesized from pyruvate in transaminase alanine aminotransaminase reaction, and this reaction is absolutely reversible. So alanine can be degraded to pyruvate. Alanine is a major gluconeogenic amino acid, so it's synthesized in many tissues. We have already mentioned that, because it is one of the ways of safe transport of nitrogen to the liver for deamination and for urea cycle. Now we are moving on to amino acid metabolism via intermediates of Krebs cycle, if you remember from our overview slides. So first, amino acids which are related to alpha ketoglutarate, and the first one is glutamate. So 5 carbon atoms of glutamate can be obtained from alpha ketoglutarate in so famous reactions of transamination or deamination. Because alpha ketoglutarate is a Krebs intermediate, so glucose can be converted in a way that it enters the Krebs cycle and of course yields to the formation of alpha ketoglutarate. That means that glutamate can be synthesized from glucose. Degradation of glutamate is reversible to the synthesis, so it can be reconverted to form glucose molecule. Glutamate is also precursor for the biosynthesis of glutamine, proline, arginine, ornithine, and glutathione molecules. So as I already mentioned, glutamine is synthesized from glutamate, and the reaction is catalyzed by glutamine synthetase, and this is one of those 3 enzymes that we have mentioned in our previous slide that can fix free ammonia. Then we have degradation of glutamine. It goes to glutamate by the enzyme glutaminase, and this one is particularly important in the kidney because upon this reaction ammonia is released, which can be secreted to tubules and which can buffer hydrogen ions originate from non-volatile amino acid, non-volatile acids, thus buffering them, and it maintains the acid-base equilibrium. And we have arginine. It can be synthesized from glutamate, via glutamate semialdehyde, and you will see the reaction a little bit later on. And then the upcoming transamination to ornithine. Ornithine enters urea cycle, as you know, and it can form arginine. But this is the pathway which yields the formation of arginine in the amounts which are adequate only for adults to replenish the already existing amounts of arginine in the body, but it's insufficient to support growth. So because of that, during the period of growth of the organism, arginine is considered to be essential amino acid. Arginine is degraded in urea cycle to urea and ornithine can catabolize to glutamate. So these are the pathway of arginine degradation. Proline, now you can see, as I've promised, the formation of glutamate semialdehyde. So glutamate is phosphorylated and then reduced to glutamate semialdehyde. So the only thing that actually changed is reduction of carboxylic group to aldehyde group, and then aldehyde group undergoes reaction with the amino group, which is the reaction of formation of Schiff base, which actually yields the spontaneous cyclization of the molecule to form pyrroline 5 carboxylate and its subsequent reduction to proline molecule. Synthesis and degradation, as you can see, they use the same intermediates but different enzymes. Hydroxyproline is synthesized upon hydroxylation after incorporation to collagen molecule, but the degradation of hydroxyproline is still unknown. Histidine is also degraded through alpha-ketoglutarate. It is essential amino acid, so we cannot synthesize it, but we can degrade it from dietary histidine by histidase to form urocanate, and this is something that we have already mentioned in a previous slide, and ammonia is released. So this was one of the source of ammonia for urea cycle. And then urocanate undergoes subsequent reaction to form 4-aminoglutamate, and 4-aminoglutamate is degraded to glutamate, and one carbon atom is incorporated in FH4 to get formyl FH4. So 5 atoms of histidine are converted to glutamate, and 1 is transferred to tetrahydrofolate. Now we're moving on to amino acid metabolism via intermediates of Krebs cycle. So but this time related to oxaloacetate. Those are aspartate and asparagine. So aspartate is synthesized by transamination from oxaloacetate, and the reaction is catalyzed by AST. This is very well known. Asparagine is synthesized from aspartate, but be careful, unlike glutamate reaction, this time donor of nitrogen atom is glutamine and not free ammonia. Degradation of asparagine is completed by asparaginase reaction, and this time ammonia is released and aspartate is formed. Amino acids which are related to fumarate from Krebs cycle, those are aspartate again, because aspartate can form fumarate in urea cycle, if you can see, and that is the formation of cytosolic fumarate. So this fumarate has to be converted to malate in order to be brought from cytosol to mitochondria, where it can enter Krebs cycle and regenerate aspartate and so on. Phenylalanine and tyrosine also can catabolize to form fumarate, and the first step is hydroxylation of phenylalanine to tyrosine, and followed by subsequent oxidation of tyrosine to acetoacetate and fumarate. So here we have the amino acids which can act both as glucogenic and ketogenic, because they form intermediates of Krebs and ketone bodies. But be careful, these are not energy generating processes. Fumarate, which is formed in this case, is converted to malate, and then by malic enzyme it can be involved in the process of gluconeogenesis following the steps and pathways that we have already explained in our previous chapters and videos. So amino acids related to succinyl coenzyme A from Krebs, those are 4 essential amino acids: valine, isoleucine, methionine, and threonine. So all of those 4 amino acids degrade to form propionyl coenzyme A, which is intermediate we got from the degradation of fatty acids with an odd number of carbon atoms, and the upcoming pathway and metabolism of propionyl coenzyme A is very well known to us. So it's transformed to succinyl coenzyme A, so which is common to their degradative pathways, and it's accomplished by carboxylation of propionyl coenzyme A to dimethylmalonyl coenzyme A, and this is biotin requiring reaction. Then dimethylmalonyl coenzyme A is racemized to L-methylmalonyl coenzyme A, and L-methylmalonyl coenzyme A is rearranged to succinyl coenzyme A in a vitamin B12 requiring reaction, and succinyl coenzyme A enters Krebs cycle. So speaking individually, methionine actually undergoes degradation through transformation to S-adenosyl methionine, and then upon the subsequent removal of methyl group, which is donated for some other processes, thus S-adenosyl homocysteine is formed, which is converted to homocysteine. Homocysteine then undergoes now very well-known reaction again with serine to form cystathionine, and cystathionine is hydrolyzed to cysteine and alpha-ketobutyrate. Homocysteine can be used for regeneration of methionine in a reaction that requires the presence of both B12 vitamin and FH4 coenzyme. Threonine is degraded by one dehydratase reaction requiring pyridoxal phosphate followed by deamination to alpha-ketobutyrate. So this is the common intermediate, which is decarboxylated to propionyl coenzyme A, and what we have already mentioned, propionyl coenzyme A is converted to succinyl coenzyme A, which enters Krebs cycle. Also, some other amino acids like isoleucine and valine can also be converted, we will see, to propionyl coenzyme A. So here we are, valine and isoleucine. Those amino acids are considered to be universal fuels. So they degrade in mitochondria of most tissues, but the highest activity of those and highest degradation of those amino acids is observed in muscles. And having in mind that approximately 25% of the average protein content are actually those amino acids, so it is obvious and easy to conclude that these amino acids are going to be catabolized in order to generate energy or to replenish intermediates of Krebs cycle. So carbon atoms of these amino acids are of course converted to succinyl coenzyme A, but this pathway starts with the reaction of transamination, after which our amino acids are converted to their adequate keto acids. And those keto acids are then subjected to oxidative decarboxylation by alpha keto acid dehydrogenase. So you have decarboxylation and you have oxidation processes at the same time, so formation of NADH coenzyme, and certain intermediates are formed. This reaction is very similar, and mechanism is very similar to that reaction of pyruvate dehydrogenase system or alpha-ketoglutarate dehydrogenase system. Then those intermediates undergo subsequent reactions which are very similar to beta oxidation reactions, yielding the formation of reduced NADH coenzyme and reduced FADH2 coenzyme. Thus, valine form propionyl coenzyme A, isoleucine can form either propionyl coenzyme A or acetyl coenzyme A. And of course, propionyl coenzyme A is converted to succinyl coenzyme A, which enters Krebs cycle. Acetyl coenzyme A can be converted to ketone bodies, so isoleucine is considered to be both glucogenic or ketogenic amino acid. One of the disorders worth mentioning here is maple syrup urine disease, which arises as a defect of branched-chain alpha-keto acid dehydrogenase enzyme, which yields and results in the accumulation of branched-chain amino acids and their alpha-keto acids. So they appear in urine, giving it the odor of maple syrup or burnt sugar. Accumulation of those alpha-keto acids of branched amino acids also results in some neurologic complications. Now let's speak about ketogenic amino acids a little bit. So amino acid metabolizing via acetyl coenzyme A or acetoacetate. 7 amino acids are considered to be ketogenic: isoleucine, phenylalanine, tyrosine, tryptophan, threonine, and leucine and lysine, which are exclusively ketogenic. Previously listed are both ketogenic and glucogenic. So phenylalanine and tyrosine are transformed in a way which is explained in a figure. So phenylalanine is hydroxylated by phenylalanine hydroxylase to tyrosine, and Bh4 coenzyme is required for this hydroxylation process, as we have already mentioned before. Then tyrosine undergoes transamination process by tyrosine aminotransferase to hydroxyphenyl pyruvate, and pyridoxal phosphate is required for this reaction. And this intermediate undergoes decarboxylation to form homogentisate, and homogentisate oxidase transforms this molecule and to several intermediates, and then the reaction is catalyzed by fumaryl acetoacetate hydrolase to finally produce fumarate and acetoacetate, which is a ketone body. Several disorders arises from a deficit of certain enzymes of this pathway, so let's list some of them. Alkaptonuria, here we are. It is the defect of homogentisate oxidase, which yields the accumulation of homogentisate, which is subjected to autooxidation, forming dark pigment which discolors the urine of infants. Later, chronic accumulation of this pigment in cartilage yields the formation of arthritic joint pain. Here we are again. This is not explained here. By transient tyrosinemia is frequently observed in premature newborn infants, and actually it's a bannic disorder which can be treated efficiently by dietary restriction. Biochemically speaking, the reason for development of this tyrosinemia is low level of 4-hydroxyphenyl pyruvate dehydrogenase. Regarding the fact that this enzyme requires ascorbate as a coenzyme, vitamin supplementation with vitamin C can help treating this problem and disorder. Let's move further on. Tyrosinemia type 2. It's the deficiency of tyrosine aminotransferase activity, which yields the formation of lesions of the eye and skin and certain neurologic problems. This disorder is treated efficiently by low tyrosine, low phenylalanine diet. Tyrosinemia type 1 or tyrosinosis arises as a consequence of deficiency of fumaryl acetoacetate hydrolase, and it tends to result in liver failure, cabbage-like body odor, and death within the first year of life. And phenylketonuria is the consequence of the deficiency of phenylalanine hydroxylase, which results in the accumulation of phenylalanine. So hyperphenylalanemia arises from this. It results in progressive neurologic symptoms and seizures, and also death within the first 2 years of life. This is also followed very often by the deficiency of Bh4, which is necessary for hydroxylation process, and this coenzyme is also involved in the biosynthesis of neurotransmitters 5-hydroxytryptophan and L-DOPA. So this phenylketonuria also results in mental retardation, and it can be treated by dietary restriction. And the point of this is the reduction of phenylalanine in the body, and also supplementation with large neutral amino acid can be applied, because those amino acids are actually competing for the same transport system, thus decreasing the reabsorption and absorption of phenylalanine from both intestine and renal tubules. And we have a little bit more, just tryptophan. It's oxidized to form alanine, and it can be metabolized, if you can see from the figure, to produce nicotinamide moieties of NAD and NADPH coenzymes. So those coenzymes can be synthesized upon the catabolism of tryptophan. And there is the rule: the higher dietary levels of tryptophan, the lower are the levels of niacin required. Deficiency of tryptophan and niacin arises in the disorder called pellagra, and the major symptoms are dermatitis, diarrhea, dementia, and finally death. And we have the third group of ketogenic amino acids. Those are threonine, isoleucine, leucine, and lysine. Some of these we have already mentioned. Threonine in the liver is converted to glycine and acetyl coenzyme A by aldolase reaction. Isoleucine to succinyl coenzyme A, this is what we have already saw on the previous slide, but at the same time it produces acetyl coenzyme A, so it's considered to be ketogenic. Leucine is converted to hydroxymethylglutaryl coenzyme A, which can be degraded to acetyl coenzyme A and acetoacetate as a ketone bodies. Lysine cannot be directly transaminated, and it undergoes rather complex degradative pathway, which involves several intermediates like saccharopine, alpha-ketoadipate, crotonyl coenzyme A, but during this process it's important that reduced NAD and FAD coenzymes are formed. The final product of degradation is acetyl coenzyme A. So anyway, that was it. I hope it was not, it was a lot, but I hope it was not that complicated. It was kind of a unique summary of our previous stories on amino acid metabolism, carb metabolism, lipid metabolism. So I hope this is going to be useful. So until the next video and until the next topic, please stay healthy, stay safe, and bye-bye.